interaction among faculty, staff, students is possible, and therefore it has been possible that you people who are having a exposure in engineering also talking about alternate fuels, and among alternate fuels the thing that is fuel which is in extensive interest in my lab, and accordingly we have developed a protocol to minimize this thing, the cost optimization of the process. And since this is a post lunch session, we'll do a little hands-on as well in order to engage the faculties, those who are there as a participants. Usually what happens is it becomes a one-way process and only the speaker talks. So this is not the objective of this particular session. And accordingly we'll also do a little audio-visual this thing also, interaction also. So we'll go to our this thing, the second slide. So what happens is waste biomass reutilization strategies is very helpful, mainly for three different reasons, which are very, very broad in terms of its application. For example, there is a potential to replace fossil fuel altogether. Then there is also an ability to recharge the groundwater level, because since you use a lot of groundwater in order to run your industry as well as your domestic purposes. So if you have a solid protocol to minimize the waste of this groundwater, so eventually you can also able to recharge the groundwater level. Then there is repairing the ozone hole as well. So if your carbon footprints are reduced, eventually the ozone holes will also get repaired, because as you know that due to various kinds of CFC and all such things, there are difficulty in the ozone layer, which is nothing but a protective against the rays sun and the other what do you call as cosmic rays. So in order to prevent all this, eventually we can have a very good output, which will reduce the burden of wastes. So here, if you see the possible thing that we can do, waste minimization. Why this? It could be done through using waste water minimization, then waste minimization, then alternative building blocks that can be used as fuels. Not directly, but through eventual processes, which we call as a downstream processing, can eventually convert those raw materials into value product like fuels. Okay? So there are several aspects to it. So for example, what happens is if we can combine the two processes together, that is waste minimization as well as bringing something which is which have noble use. So in my case, in our case, we are trying to treat the wastewater and eventually try to capitalize on biofuels also. So we'll do a little this thing also. So what happens is, as you can see these days, which is mainly a civil engineering problem, okay, or environmental engineering problem. The problem is there are huge wastes that is being generated either from domestic sources, either from municipal waste or from industrial waste. Okay, so you will see that nobody is interested in waste reutilization. There are some interest from some government agencies and some NGOs. Are you getting my point? What I am trying to say is, for example, there is a huge pollution happening everywhere in all water bodies. Okay, so government is doing its part, trying to do at least, but this thing, what you call as private players are not there. Why private players are not there? Because the benefit is not instant. Understood? If you put a wastewater treatment plant, it will be not a beneficial aspect. Therefore, large industries are not coming. So how can you encourage a large industries to come in wastewater treatment? So in order to do so, you have to combine this wastewater treatment protocol with that of something which can produce high value products. Are you getting? For example, biofuel. Now in biofuel, there are several strategies that are there. You may also come across several other strategies to develop biofuel. So generally, what happens is I am a biomedical scientist, and my profuse interest is in biomedical applications. Okay, so what we have understood in our lab is there are certain microorganisms which can accumulate biofuel in their cytoplasm. Okay, this is audible or not audible? Hello.
Yes, sir.
Is audible?
Audible, sir. Continue.
Okay. Audible and visible, right? Visible also.
Also, sir.
The screen. Okay, very good. So okay. So what happens is what happens is this our process is bit different. In our process, what we are doing is we are eventually recycling the wastewater. Okay. At the same time, we are also producing biofuels by using microbial cells. Now, in microbial cells, what happens? There's each and every microbial cell have some specific. No, it cannot be maximized more than that. Achal, let me see if it can be done. I'll just see. Okay. Yes, it can be done. Now it is better, I think. Is it better now?
Yes, sir. Yes, sir.
Okay. So what has happened is, so what we are trying to do is we are combining two processes together using microbial cells. Now microbial cells are nothing but microscopic cells which are already there in the environment, but we need to analyze them, isolate them, and so that we can standardize the process so that through microbial cells we can generate biofuel. Now, during this biofuel production, I tell you very simple chemical engineering process, which is called esterification. In esterification, what happens is you combine one molecule of glycerol and three molecules of fatty acids to bring about one molecule of what we call ester. Now, this ester is nothing but the biodiesel or biofuel. Okay. So what happens is now these microbial cells are nothing but living cells only. Understood? Now these living cells also require what you call as some source of food, isn't it? Some source of nutrient. The wastewater itself becomes a nutrient. Understood? Because in wastewater, what happens is consider that you are taking wastewater from domestic sewage. In domestic sewage, in the wastewater, there are plenty of molecules which are can be used by the microorganism as a source of nutrient. Understood? So what they'll do is they'll accumulate the source of nutrient from the wastewater and eventually they'll convert the nutrient into biofuel. Okay, the biofuel chemistry is there in any organisms. Once organisms we have isolated. Okay, we will come to those details. Now, what is the benefit? The benefit is that you are adding some value to the system because of waste you are producing fuel. And there are specific protocols for this. It is not so easy that the way we are talking it. It is not easy to execute. It needs a lot of skill as well as infrastructure in the lab. So we'll come to those points now. So I will give you that since it is already a transition, people may feel dizzy. So I'll request the coordinator to play the first video. I have shared some video links. So please play the first video.
Sir.
Share your screen and play the first video. Yes, share your screen and play the first video.
And also, sir.
So, hello.
Sir, same, sir.
Yes. Okay.
Innovation.
Very good.
With population and income.
Okay. Now here you see what happens is a lot of innovation. Yes. No. Sound off. Mutation. Mute. Yes. Maximize the screen. Okay. Now what happens is innovation is going on everywhere. Okay. It is like a prey-predator relationship. Now these are organizations which are not depending on biofuel. Okay. They are asking for more and more what do you call as conventional fuel, that is fossil fuel. Now in order to, since what has happened is fossil fuel is reducing day by day, everyone knows. Now these organizations, what they are doing, they are using a lot of innovation. They are bringing about the innovation landscape where they are doing everything to accumulate the last drop of fossil fuel. Now here you will see what they are doing. Now they are organizing, they are putting a whole ecosystem there. They are engaging scientists, labs, institutes, industries, and everywhere, so that they'll generate intellectual property in order to find out whatever fossil fuel is left there in the earth. Are you getting? So in order to do so, what exactly they are doing? See, so they are organizing this thing, networking to all kinds of intellectual people out there. Okay, and then they eventually have developed a protocol where they are using sound waves to find out this thing, fossil fuel. Okay, now this is the whole lot of ecosystem they have generated. Okay, so how they'll do it? What is the innovation? See, now these are some vehicles. From these vehicles, they'll generate sound waves or radio waves. Okay, So according to the analysis of these radio waves, what they lot this may be seismic waves also. So on the basis of the signal processing through some protocol, what they'll find out is, and they are doing it in the sea also. What they'll find out is they'll find out where the fossil fuel is trapped. Okay, it may be very deep inside. Okay, so in order to procure them, what they'll do, they'll analyze a whole lot of data. In order to do pinpoint a very small thing from a large haystack, you need to analyze a lot of data. So after several data analysis, they'll find out where exactly the fossil fuel is left. Now, in order to procure the fossil fuel, what they'll do, they'll deep inside, they'll dig into, and then they'll put a lot of brine, that is hot salt water, and then they'll pump it so that it will come out along with the brine, that is the salt water. The petroleum will come out, and then eventually they'll distillize it and then put it into a bottle and use it for various purposes. Okay, including jet fuel and all such things. Okay, so this is how they are doing the innovation. So why you are trying to say this? Okay, close it. So why you are trying to say this? It is like a prey-predator relationship. There are two classes of scientists. For example, in our lab, we are trying to find out some rational and some what do you call it, sustainable process in which we can bring about some real change, which will limit the use of what we call as fossil fuel. Okay, so the one more group of people are there who are not looking into it. They are trying to utilize existing technology in different way. Okay, for example, I give you, for example, you have procured a Tesla car, but you are charging your Tesla by using, hello, is it audible? Is it audible? Hello, is it audible?
Audible.
Okay, if it is audible, rest of the people switch off your mics. It is very difficult. It gives a little difficulty. So what happens is, there is a prey-predator kind of relationship. Okay, now what is a prey-predator kind of relationship? For example, the deer is moving fast. Okay, behind the deer there is a tiger. The tiger want to eat the deer. So evolution is taking place in both way. So it is changing the behavior of this thing also, the deer also, and therefore the deer is getting rid of the bigger horns it had. Otherwise, it will get trapped in the forest, and the tiger will come and eat it. Okay, so what is happening? The deer is changing, and it is changing its the structure of the horns. At the same time, the tiger is also changing. Previously, tiger was able to move like 5 kilometers per 5 minutes. Now it is moving 10 kilometers. Okay, so in this way, what is happening? This evolution is taking place for everyone. So the scientists, the group of scientists that we are trying to some kind of, it's definitely an interdisciplinary kind of environment where biotechnologists, biomedical scientists, and engineers will come join in hand with mechanical engineers, then environmental engineers as well, to bring about a real change, isn't it? At the same time, the other way around is also true. They are also trying to use the fossil fuel strategy, isn't it? Because it is already a lucrative business, isn't it? So there are different these things are there. So I'll go back to my this thing session in my this thing presentation. So you can see it. Then okay. Okay. So there are plenty of other things as well. So as I have told you that what exactly we want to do, isn't it? So how exactly we will do it? So this is the protocol which has been developed by Helsinki or Finland-based institute, where they have combined the both of the process. They are doing this thing recycling, water recycling, as well as what do you call as biodiesel production. Okay. So here they are putting a different kind of microbial strain. We are putting a different kind of microbial strain, but we are trying to say that it is possible, and then you can make wastewater treatment a lucrative business, because you have hybridized your wastewater treatment plant with something beneficial, that is biodiesel. Now one more thing is there, biofuel concept. So you can ask me, sir, why the biofuels are not available in the market? There are several bottlenecks, technical bottlenecks. One such bottleneck that you are already aware of is the structure of the combustion engine. So you cannot directly use the biofuel directly into your existing combustion system. In order to do so, you need to change some these things, upgrade or like modify your existing combustion system, isn't it? So this is one bottleneck. There are several other bottleneck also. But one thing is very sure that within the existing technology we can use the biofuel or bio oil. Okay, that means completely biogenic oil or fat directly into a system which will helpful for your friction reduction. Okay, so what happens is today we have the technology to make a lot of this thing what you call lubricant, which is also a big industry. Although it is not fuel directly, but it is also have a lot of thing to do with the same industry, that is the mechanical and automobile industry, isn't it? So today also whatever the existing lubricants are there, these are mainly made up of plant-based oils as well as some what you call is mineral oil. Okay, so this can be directly replaced by biofuel. That is possible by microbial engineering. So we shall show you this. So one more bottleneck is the downstream processing. Now what is the downstream processing? Downstream processing is very simple to understand for mechanical engineers. In what happens is we are developing a growth culture, okay, where your microbial cells are growing. Now these microbial cells are very difficult to separate from the tank. Why? Because it remains suspended throughout the tank. They do not make three-dimensional floccules that you can filter it out easily. Understood? So this is again a bottleneck, and for that we have to rely on a process called this thing. What we call is bioprocess technology. In bioprocess technology, what we do is we do a process where it becomes easier to separate the microbial cells. So as all of you can understand, microbial cells are very small. A single cell is so small that you cannot see by your naked eye. Now, if this cell produces biofuel inside the cell, so you have to release the biofuel also in order to use it. Okay, so it may be difficult for you to visualize initially, but later on you can understand the concept. The concept is that it is a single cell which can produce biofuel and which will be entrapped into the cell. So if it is already entrapped into the cell, so what will happen is you cannot separate it easily. So in order to separate it, what you have to do is you have to put this thing as proper kind of centrifugation. Now centrifugation is also a very high energy demanding process. We shall come to this point. So in this slide, what we are trying to show you is recently after 2000 there has been rapid development in bioprocess technology, and therefore now we are having some technology that we can use to reduce the cost of bioprocess or to reduce the cost of processing the biofuel from its basic nature. Okay, so we'll come to this point. So these are different aspects to it. There are these are the compounds which may be present. Just hold on. Okay, so these are the compounds that can be used by the microorganism from wastewater, and it will get converted into biodiesel. So we have standardized the technology. So what are the waste that can be present? This can be organic waste, this can be glycerol, this can be fats, and this can be alternate polysaccharides, which are all present in any kind of waste if you see. Okay, in your domestic waste, industrial waste, all waste contain all these things. Now, any one of these can be useful for the organism. That is microorganism we are talking about. This particular microorganism, what it will do is it will take this waste material from the wastewater and then convert it into biodiesel. Are you getting? So we'll also come to this. We'll now see at the second video that what exactly is going on. Okay, so I'll request the coordinator to play the second video. Okay, so this is the conventional fuel which can be used for everything: automobile, large vehicles, aeroplanes, then all other big equipments. Okay. Similarly, yeah, all kinds of possible ways usage of oil, conventional oil. Similarly, biodiesel can also be used for all this, but the technology is little far away. Okay, not exactly ready. Okay, so this oil is procured from beneath the land, as you know, and the cost is increasing day by day. Okay, but there is an advantage of biodiesel. In biodiesel, see, the dollar price is increasing. That means the cost per what do you call barrel of this thing petroleum oil is increasing day by day. But in contrary, the biodiesel the cost is not increasing in a rapid way. Why? Because the raw materials are very cheap. One, sunlight. Second, carbon dioxide. Three is what? The nutrients. As I have said, the nutrients, from where the nutrients will come? From the wastewater. So are you getting? So all the requirement, the raw materials are very cheap, and therefore the biodiesel overall will be very cheap. But still, it is very costly because the downstream processing is costly. I'll come to that point. Now this is a microplate. It's a petri plate where the organism is growing. Okay, from that organism only you will find biodiesel inside the cell. As you can see, inside the cell only all the biodiesel is trapped. Now in industry, how it will look like? Now it will look like this. Can you see? These are the fermenters. Large big boxes are there. These are all fermenters. Okay. Now in this fermenter, three kinds of things are getting in. As you can see, the carbon dioxide is getting in. Then you are putting the nutrient as well as the a particular kind of strain. Okay, we'll show you. Just hold on. And when we mix all this in a particular temperature, particular pH, particular ionic environment, aeration, oxygen concentration, and several other things, you get a flourishing growth of biodiesel. Okay, here you see. Okay, so what are the happening? All the things are getting mixed inside the reactor. Now the reactor is laden with several sensors. Now they will understand everything: pH, ionic component, temperature, and all such things. Okay, and accordingly it will change also. If the pH need to be adjusted, a buffering system will be activated, and whenever temperature is change is required, and then a temperature monitoring system will be deployed, and eventually it will become very feasible. Understood? And the cost of the oil is also very low. But with the development of technology, we can use the biofuel in every aspect of our life. Understood? So there is a huge potential that eventually we can replace it. Okay. So you close it. I'll share my slide. Okay. So what has happened is, okay, now these are some production strategy where we are trying to show you how exactly various kinds of biodiesel. Okay, one such example is EPA, eicosapent pantanoic acid. Okay, it's a fatty acid only, and it is having a great potential to become a biodiesel because its octane number is very good, and it can eventually become a fuel also. Right now, we can use it for this thing what we call is lubricant. Okay, at the lubricant which you can directly add into your combustion system. Okay, if you can remember, in your scooter you used to add the lubricant directly in the combustion system, isn't it? So similarly it will be acting, and this is the process through which we can generate it. So I am not going to do the detail because it is all chemical engineering and production engineering. Eventually what will happen is, as you can see, there is a lot usage of centrifugation. Understood? Because centrifugation is a very basic fundamental equipment which is useful, but it triggers a lot of energy. Understood. So in order to produce any kind of biodiesel, you are highly dependent on centrifugation. So I'll give you some more this thing usage or more applications where centrifugation becomes what do you call indispensable in biodiesel industry. Okay. So here again, we are giving you one more this thing. It is also for specific interest for those who are having a specific interest in biodiesel production and its chemistry. So here also, as you can see here, centrifugation is must for several other steps. The initial steps are like this. We will do this thing what we call is the microalgae based process where we will do the cultivation, then harvesting, dewatering, then lipid extraction. Lipid is nothing but oil. Okay. So this oil is because it is biodiesel, therefore it is the chemistry is not exactly same as the petroleum molecule, okay, which are highly volatile in nature. This is also volatile, but the chemistry is a bit different. These are generally long chain fatty acids. Okay, Their octane number is very good. They are very, but not as inflammable like this. But with some specific adjustment in the combustion system, we can definitely able to use it. The way we are putting alcohol these days, okay, isn't it? In Brazil, what they are doing is they are putting a lot of alcohol in the petrol directly. Understood, and therefore they are using a hybrid kind of fuel because ethanol is very cheaper than that of petroleum in those countries because they have a large amount of sugar cane, then malt, and many other things which can produce cheap ethanol. But in India, the case is not like that. So in India, we can definitely cash on biodiesel. Okay. So here also, as you can see, a lot of use of this thing, centrifugation. And due to the use of centrifugation only, what will happen is we have done a cost balance analysis where we have seen that due to the use of what we call is centrifugation, the cost increases rapidly. Okay, so here also again one more strategy. If you have specific interest, it will be very interesting to you. Otherwise, since it's this thing, different kinds of participants are there, I'm not discussing it much. But here also, the usage of centrifugation is very needed. This is again a very interesting case. Recently has been published in the Journal of Fuel, an institute which is in what do you call as Middle East. Okay, so Middle East, in one hand, they are trying to cash on the existing fossil fuel reserve. At the same time, they are also trying to do innovation where they are trying to explore the opportunity in what do you call is biodiesel. Okay, so why? Because in Middle East there are huge open barren land which is of no use, but a lot of sunlight falls on them. So what they are trying to do is they are trying to capitalize on the sunlight, isn't it? For biodiesel production, you require a huge amount of sunlight because it's a autotrophic or phototrophic reaction where sunlight is being used to produce biodiesel. Okay. So this is the chemistry where they have isolated a particular kind of bio oil from microbial sources. Okay, I'm not going to discuss it in a detail. So here also this thing of centrifugation, each and every step of biodiesel depends on centrifugation. It starts from the beginning. It starts from the yield of the microorganism. You think like this, that you are having a pond. Okay, many times you will see ponds where the water becomes completely green. How many of you have seen ponds like this? Participants who are already teachers. So they are students. They themselves might have seen ponds in many ways. Mainly those people who belong to a rural setup like myself. I belong to a very small institute. I'm sure many of you, nobody in this particular session belongs to more smaller institute like mine. But despite of this, we are very much this thing in R&D process within our limitations, and we have generated sufficient amount of intellectual property in the form of research papers and then patents as well. And we are trying to market our patent also. And we have had interaction with some small scale industry who have shown specific interest to our protocols. Anyway, so what happens is you can see many ponds which are completely green in color. Why? Because it is full of algae. Now in that kind of pond, you see from top to bottom, the whole system is fully green. Why? Because the microorganisms are such that it remains suspended. It do not get precipitated. Understood? Understood. For example, in order to precipitate a dirty water, what you will do? You will either do precipitation, or you will add some kind of electrolytes. Okay, divalent electrolytes like aluminum, and then the impurity will be precipitated. But in this case, you cannot precipitate it by simple process like this. You need to have specialized membrane filters or some specialized what you call centrifugation. The algae generates nicely, but it is difficult to accumulate it. Understood? So, and therefore you need to invest a lot on your centrifugation process. Now these are the centrifugation process everyone knows, and there are some pros and cons, but these are the different centrifugation system which are still in use in biodiesel industry. Okay, and eventually we have able to find out the cost of this thing also, centrifugation also, and therefore it has also been able to, we have also been able to calculate the cost of per unit biodiesel production only due to the this thing what you call centrifugation process. As you can see here, the energy balance of the centrifugation process can be calculated using equation number 1 to 3, assuming no loss occurred during the downstream process. The energy input for unit process kilowatt hour per 1000 kgs of dry microalgae, and that can be calculated using the following equation: ELX is equal to VX into EX. Okay, where Vx is the volume of slurry and X is 1, 2, 3 like this in cubic meters, and Ex is the energy input for unit process X, that is kilowatt hour per 1000 kgs. So eventually you can able to calculate this kind of energy investment that is there in order to process 1000 kilos of what you call is biofuel that is coming directly from microalgae. Okay, specific technique, not any kind of like jethropa and many other things are there. But we are only interested in the microbial source of this thing what you call is biofuel. Okay, so here I can just hold on. So here what we have done is there are specific varieties of microalgae. Okay, let me tell you. So microalgae is what many you may not understand. Microalgae is nothing but the same organism which makes your pond water green. Understood? So in our lab, what we have done is we have checked n numbers of ponds, and then we isolated a particular kind of microalgae which is good to produce what we call as biodiesels. Okay, which will be accumulated in the cytoplasm. Understood? So there are n number of varieties, many varieties which can produce it. So there has been a report where on the basis of different varieties of microalgae, they have calculated the amount of energy that need to be invested. Okay, this is also for specific interest only. Okay, now centrifugation. What are the pros and cons of centrifugation? The disadvantages are huge. It is very costly, time consuming. Time consuming, that then there are large initial capital investment as well, then high labor equipment is also there, then high operational cost is also there, high energy requirement is also there. But in a nutshell, it's a very huge expenditure. Okay, so what will happen if we can directly remove this particular this thing by some low energy investments? So it will drastically reduce your biofuel production system. Okay, so these are also some costs. Scale up information. Now in scale up information, as you can see, for centrifugation alone, the maximum capacity and size, if we want to reach, we need to invest a lot. It is 2,50,000 euros in order to scale it. Okay, in the largest possible way. If you want to scale up for a centrifugation for a maximum possible way, so you need to invest a lot. So in order to reduce the cost, there must be some way out. So we shall find out what exactly are those. And we are also doing a lot of brainstorming with some other institutes and industry as well to do some valuable research in this particular direction. Anyway, in our lab, what we do, we find out from the natural sources different types of microorganisms, microalgae, which are able to what you call as use as a biodiesel, because all microorganisms will not do. Some specific organisms will do. Some organisms will be this thing. They'll generate microalgae, but they'll generate biodiesel, but it is of no use. But in our case, we have isolated. We are trying to find out a particular this thing, which will eventually become very useful for initially for lubrication purpose. Okay, so we'll share a video also, not ours, some other institutes which also doing the same way, but with a different kind of strain. Anyway, now this is the basic protocol where we are trying to isolate a particular strain from the nature and then put it into the lab and doing all kinds of analysis, lab based analysis, and eventually we will put it into the industry to scale it up, and we will see what will happen, what will be the behavior of the organism in industrial scale. It may happen that in your lab you are getting very good results, eventually in the industry you are not getting it. So eventually all your hard work will be of no use. isn't it? What is your objective? Objective is to generate intellectual property, write a letter, write a paper, submitting a patent, but eventually you also have to monetize on it. If it is of no use, nobody will read your paper, isn't it? So eventually what we will do is, now this is the growth kinetics of the organism. So it is also very important, but also very interesting only for those who have specific interest in this. Anyway, given an opportunity, I'll discuss it. So what has happened is all organisms have some specific growth curve, okay, including human being. Human being also develop in at a certain point rapidly, then slowly, then start decaying and eventually dies. So microorganisms also do like this only. And it has been found that during the intermediate phase, can you see 4 different phases? Log phase, then lag phase, and then there is a stationary phase and then a decline phase. Okay, so it has been seen that during the stationary phase. Now, what is the stationary phase? The stationary phase is the phase where the organism is very comfortable. Initially, in the lag phase, the organism is not very comfortable because it has come to a new environment. It is trying to find out what exactly is going on everywhere. Then eventually, when it finds out everything is fine, it rapidly grows. Okay, the number of the organism rapidly grows 2 to 4, 4 to 8, 8 to 16 likewise. Now we call it log phase, logarithmic growth phase. Here also no exterior activity takes place. Now after the log phase, there is a stationary phase. In stationary phase, what happens is whatever exterior activity is there, it will do. So initially the organism's objective is to increase its number. It is also like human being. What is the overall objective of human being? The overall objective of the human being is to grow the number of its own progeny, isn't it? So eventually the bacteria, the organism also acts in this way. Initially it will only grow in number and later on it will try to produce the additional things. Now that additional thing is our thing of interest, that is biodiesel, which will be accumulated inside the microorganism cell. Okay, then you have to liberate it, then put it into a packet and then you can sell it. Okay, and eventually there is a decline phase. So in order to understand the chemistry, we also have to understand the growth nature or growth kinetics of the organism. Okay, so this is the growth kinetics. Initially from 2 to 4, 4 to 8 it will grow, and the organism growth and decay in a waste recycling process in industrial reactor is given by dx by dt equal to mu s into x into I and then kd into x, where x indicates the microbial biomass concentration in gram per liter. The term mu s is the substrate concentration dependent specific growth rate. Then mu can be further defined as mu equal to mu x into s by ks plus s, where maximal specific growth rate is mu x into 1 by d, and then substrate concentration is s in gram per liter. Another term ks is the half saturation constant. Now I is the product inhibitory term. Inhibitory term means what? Inhibitory term means something which will inhibit the growth of the organism. So all positive and negative, all kinds of things, because you are giving sufficient nutrient. It may happen in the nutrient there are some toxins which will kill the organism. So you have to bring about. You should also talk about that thing also. Everything will not go as you planned, isn't it? In all your labs, whether your mechanical lab or any kind of other lab also, whenever you are trying to do something new, you face a lot of problem. So it is better to equip beforehand only. Before executing the whole experiment, you should also think of what can go wrong. So eventually there are several other things. I am not discussing it in much detail. So the organism growth and decay in waste recycling process in lab scale also we can estimate and count, which is very, very important for the simulation purpose. So otherwise what will happen? You have done everything and eventually you see that nothing is happening. Now this is a basic. What do you call is this thing? What do you call is a tool in which you can grow your microalgae. Okay, so this is again for mechanical engineers also. They can devise a more better device which will give a sufficient specific surface area to grow the microorganism in the presence of sunlight. If you can see, there are like in the number 1, the loops are there. These loops are made up of transparent material, so that what will happen is your sunlight can be there. Understood? The sunlight can be there, and sunlight can help to grow the microorganism, isn't it? So if you can grow sufficient amount of microorganism, then you can regulate the growth also, and eventually once a lot of microorganism has been grown, you can separate them by using centrifugation. The centrifugation system is not shown here. It is only the suspension. The suspension you need to put it into the centrifugation and then you can concentrate, isn't it? You have to evaporate the water, and so that only the cells will be get concentrated. Okay, so in order to again give you some more or this thing discussion on it or more insight on it, I'll request the coordinator to share the third video. Go ahead, third video, share.
At the end of the day, you produce high-value products which can contribute positively to the market. The energy to grow algae
Mute it.
free because
Mute it.
light from the sun. And
Mute it.
as we will
You mute it. Let it play, but mute it. I have written in specific this thing in email. So what has happened is this is the pond where a lot of exposure. Can you remember the plant we have shown just now? The smaller device. It is nothing but a smaller version of this only. Okay. So can you remember the plates we have shown? Okay. The pond they are using the whole pond. Okay. You can do a miniature version of it also. Now this is the lab. Okay. In the lab, what they are doing? See, this is a very interesting system. It is not a basic microscope. It is we are calling it FACS, fluorescent activated cell sorter. Each and every cell will be analyzed. Understood? Each and every microorganism should be analyzed so that you get a uniform kind of variety. Otherwise, what will happen? Some other microorganisms are also growing. So in one cell, there will be 25% content of biodiesel. In another, there will be 75. So eventually, you will lose your uniformity. So there are several aspects to it. So this is the pilot scale this thing only system only where here you see the FSCS. Each and every cell will be passed from a laser beam, and then if the uniformity is not there, we will reject the batch. If the uniformity is there, then you can what do you call continue with the batch. Okay. So what happens is the algae, as I have said, they use sunlight, carbon dioxide, and nutrients. Now this nutrient we are trying to replace it with what you know wastewater. Understood? If you put some good thing in that, so what will happen? Again, your cost will increase. So there is a potential, huge potential that you use wastewater for production of this kind of thing that these people are doing. These people are doing it for a IOS. IOS kind like what I'm trying to say. I could not show you this thing. In another video, they have shown you the packets. In packets, they are putting this microalgae and using it for lubrication purpose. Okay, that video I did not share. I also had that video anyway. So what happens is it is there. The existing technology is there, but it becomes very costly when you are using what high value products or high value raw material. If your raw materials are cheap, then eventually your final product will also be cheap. Now that is the objective. But there are several problems in it. The problems, as I have said, to accumulate the biomass. The biomass will be grown. Like in many pond, you will see that on the top of the surface layer, there are so many organisms, plants, and then big trees also, isn't it? So that you can separately easily. But the suspended material you cannot able to separate easily. Understood? It always remains suspended as a liquid, but you need to make it like accumulate all the things so that all the cells are together, and then you can accumulate the fat which is trapped inside the cell. Okay, a microbial cell is nothing but something like a round. Inside the round, the oil is there. Outside the round, there is no oil. So you have to accumulate all the round cells together, and then you can use it, isn't it? So okay, so we'll go again. Okay, here again, just hold on. Okay, can you remember the the pond the person showed you in the previous video, it is like similar to this one only, isn't it? Anyway, there are some adjustment that has been done because it is a lab scale thing. So here again, there are some this thing, the cost, overall cost of the equipment that will be needed for your biodiesel plant if you are using the strategy. So in that, what happens is you need to put centrifuge all over again. Can you see delivered cost per dollar? Can you see the number two centrifugation? How much it cost? $1,23,949. Okay, the overall cost is $2,47,899.98. Okay, percentage of MEC, the total cost is approximately 28%. The overall 100 out of 100, the 28% cost goes to centrifugation. So we must definitely see to a point where you can minimize it. Okay, so that we are doing in our lab in a very significant way. Okay, now due to this only, the biodiesel market is not very widely this thing. True biodiesel, I'm not talking about only the putting alcohol in petrol. It is also a biodiesel, there is no doubt in that, but we are talking about completely generated diesel, which is also still a far cry. It is not very easy to procure it right away. So what we can do? What we can do is we can definitely look into it, that from microbe developed biodiesel or biofuel, we can put it and use it as a lubricant. Lubrication industry is also very big, and it also uses a lot of this thing petroleum product as well as some plant based. Because all of you are very much expert in mechanical engineering and combustion system, you will definitely know, in order to reduce the friction, this plant oil is very good, and most of the lubricant that is there in the market uses this thing, what you call is the plant based material, isn't it? So in this way, what happens is you can definitely have a process where what you can do is you can directly go into a process where complete biodiesel microbial based thing you can use for your existing system. Okay, you need not to change your combustion system altogether, because you are not putting it directly into the combustion system as far. Many times you are putting in some other ways, isn't it? Not inside the system. So what will happen? You can definitely get what do you call is an insight to use lubricants for plant based or microbe based lubricant. That is a true biodiesel. So how exactly we can do it? So the green dots, can you see? These are nothing but the cells where inside the biodiesel is there. Okay. So the problem is that they are not getting accumulated, and therefore you need to put centrifugation. So what we can do? We can artificially coagulate them. Are you getting my point? So there are two ways. We can use the fungi. Now there are two different kinds of fungi. Can you see? One kind of fungi produces long threads. So in between the long threads, the microalgae will get trapped. Okay, but one more type of fungi is that which are not filamentous, they are small fungi. Now these fungi also can able to what put a matrix in between the suspended cells. Are you getting the what is the problem? The problem is that the microalgae is not getting coagulated, they remain suspended in the water, and the water becomes completely green in color, isn't it? So in order to reduce this, what you can do is you can use microalgae. So the algae, what it will, the algae will either produce this thing hyphae, and therefore can able to filter it out. Otherwise what it can do is it can also understood. So this is how we can this thing eventually into a different. So what here we are going to do is we are going to put the strategy into the cell itself. In the before slide, you see we are using one more organism. Can you see? One is fungi, one is algae. Can you able to see what into microbe system? In order to make different organisms the same suspension, it becomes very challenging. One microorganism can grow with waste, but the rest of the other microorganism cannot grow on the So it becomes challenging for you. So what we are trying to do, please listen to me. We are putting the characteristics of one organism into another through a process called genetic engineering. Okay, now what is genetic engineering? Genetic engineering is something like the putting the characteristics of one organism into another. Okay, and literally we can able to do it. Okay, now here you see. So this is the molecular mechanism of accumulation. So this is nothing but yeast. Yeast, you know, yeast is nothing but the organism that you put for your card, that you also put to make your pizza dough. The pizza you have on that one round thing comes. Now it comes from a dough. Usually you use yeast for your idli as well as for dosa. Fermentation of the batter. Understood. Now this organism is coagulating. So what it does is whenever it grows, it forms full or large mass of cell. It do not remain suspended. Understood. But the usual thing that we are talking about, organism which is cell, will not become like this. It will remain suspended only like this. The green dots. Understood. What we are trying to do is we are trying to see how these yeasts are clumping with each other. So we found out that in between the yeast cells, there is a thing called flow, FLO. Flow is flocculation enzyme protein. Understood. Now this protein is specific to this yeast only. So what we are trying to do is we are trying to put this flow protein inside these organisms. The green dots behave like this, double dots. Okay. So what will happen? Your centrifugation, if it will not be much extensive, a small amount of centrifugation can help it, or simply you can suspend it. Like if you keep for a long period of time, they will suspend all by themselves due to gravity. Just hold on. Okay. So what we are trying to do, we are trying to do a kind of completely biomedical, hardcore biomedical research. And before going to that, for general understanding and to your students that this kind of innovation is going on, because at the end of the day, the major work of the teachers is to teach their students, and we must equip our students with advanced knowledge that is already there. So you can definitely able to talk to your students that this kind of technology is available, and we are exploring this what we call as this particular thing. Okay, now this is the molecular mechanism how these yeast are doing it. In yeast, what happens is there is a particular kind of gene which produces these plugs. Can you see the blue color plugs? These plugs are nothing but the protein that is there. So what they do is these plugs starts to club each and every cell together, and therefore they make a large clump. Okay, the clumping is possible. So what we are trying to do is we are putting the genes of this plug protein, okay, or FLO protein, flocculation enhancing protein, and then putting it into our microalgae. So in order to do so, we also have to find out which kind of flow is needed. As you can see here, there are various types of flow. Okay, all they look same, but at molecular level they are not same. So it is also a challenge that which particular organism you will, which particular gene, because there are huge amount of genes. Okay, so if every time we take one gene and put to analysis, it will become a very big burden. So we have to also understand that which will not work and which will work. So in order to differentiate between the false positives and false negatives, we have to find out the exact true positives or true negatives, isn't it? So in order to do so, we can do a little simulation here. Okay. So what happens is I had shared some files, but I'm not very sure that whether it will be of any interest to you because most of you are mechanical engineers. The molecular mechanics concept of proteins is not very relevant for you. However, since given an opportunity, I'll try to explain. So what has happened is this is how we are standardizing the protein. Okay, which protein or which flow, because there are plenty of flow. Some flow means nothing but the plugs. So what will happen is, please understand, when you are encouraging the yeast to grow, okay, for making curd for you, making pizza dough for you, or for making the dosa batter or the idli batter, you are giving it a very good environment, okay, which is needed for these organisms. But when you are using wastewater for your food, not the pizza butter. Understand? So what happens? There are lot many more challenges. So you have to be very very sure which is more stable plug. For example, you are using a plug your experiment is of no use. So you have to find out which plug is very very stable under high ionic stress, because in the wastewater it is not a controlled environment like your dosa batter or your what do you call is this thing what do you call your curd. Okay, now curd is a settled thing and there is no dynamic movement in it. But in wastewater there are so many changes. You cannot predict the structure of the wastewater, isn't it? So you need to find out a very very stable plug. All the plugs you cannot use. Only one plug you can use, which is best stable. So in order to find out the stability, we can do a simulation. Now this kind of simulation at molecular level or molecular mechanics, we call it molecular dynamics simulation. Okay, for material scientists also it is very important. So in that what we do, we are putting several stress and trying to see what is the effect of the stress on the molecule. Okay, for example, you are also using some kind of simulation tool, isn't it, to find out the stress on a particular material or to find out the Young modulus, or to see the how the tensile strength of a material, how good the this thing the heat transfer capacity of a material, this and that. So many simulations you must do. So we are also doing a kind of simulation. We are trying to say that how much good is the so what happens is we are trying to simulate what exactly is going on and to find out the best possible way to understand which protein is good for this. Okay. So in order to do so, I'll request the coordinator to play the last two videos simultaneously so that they can understand which was his better protein. Okay. So we have shared with you two more last videos. If you can use two different players, you can run both the files together. Yes, these two files we are talking about. Use two different players for it. Minimize it. Minimize it in a small window. You do two different. Echo in one you use one kind of player. In another you use another kind of player. So both the videos you can play, and simultaneously our participants will understand what is stability and which is instability. If I show them different way, and minimize it again. Try to minimize it if it is possible. See if it is possible or not.
It's not for you. What do they
It could have been possible. No, you could have done it. Anyway, leave it. So what happens is, now see, this is one plug. Okay, we have done the simulations in a particular constant temperature and pressure, ionic environment and ligand. We have used the one first plug. Now, first plug, you see, only you can see only a small part of it. The whole structure you cannot see. Why? Because it is very, very fragile. Under stress, it becomes completely fragile. So you cannot use this plug. Okay. What is your objective? Your objective is to make suspended material, suspended cell into a flocculating cell, so that by gravitational force only you can separate the biomass and put it into a packet in order to make the this thing lubricant, isn't it? Microbe based lubricant. but you have to definitely use a plug which is very stable even under wastewater environment. Are you getting? So in order to find out a good plug, there are several classes of plug. For example, flow 1, flow 5, flow 8, like this. Understood? These are all flocculating enhancing protein which is present in yeast. Okay, so out of the yeast material, only one you can find out which is most stable. So this is less stable. Okay, so the second video you play. You play the second video. Here you see. Can you see? It is also fragile with the same what you call simulation box only. In the simulation box, you can able to regulate the temperature, pressure, ionic force, shear strain, shear, all kinds of thing you can put. Okay, so we have put the same thing on two different proteins. In this protein, you see, it is also getting broken, isn't it? It is also breaking down, but the extensive breakage is not there. Can you see? You can see the helical parts. Can you able to see the helical structure? No, it is nothing but the protein or the molecular level structure of the plug. Okay. So what does it mean? It means that under same molecular dynamics parameters, the second protein is more stable than the first protein. Getting my point? Therefore, we have taken the first protein only. Otherwise, what we have done? We would have done all the 8 different flow proteins are there. Every day we have to do genetic engineering and eventually we will fail. Now we know due to the presence of the very good simulation tool, now we know which flow protein is better. Understood? Okay. Anyway, it becomes highly interdisciplinary, but I hope that it will give you an insight. Anyway, thank you for this. So we'll get back to our own thing. Okay. So eventually, the whole analysis and all sort of things that we are expert in, we have done in our lab. The object identify a bug protein which is very stable and eventually it helps help to the organism is there. The problem is that you cannot precipitate them. Just hold on. I think you have to unshare your screen so that I not able to. I'm saying. Can- Okay, now this is the strategy. Other things which is completely okay. Now these are the proteins in of specific interest. That these are the three different even one. Okay, molecular and for mechanical engineering. There's a whole lot of stuff that we use for developing this protocol. Find out is good. Self which will to move the to strategy understand a particular time will be more for us and the report screening
Sir, it audio is not coming, sir. Sir, share your.
Uh,
Sir, your side is not on. Just check it.
it is not getting shared. Na? I don't know why.
Audio is
Till
clear.
now it's
No
fine.
video. Your PPT is not saving. Just coming up.
Okay, okay. I see, I see. I'll once again try. Can share the screen. I come to the only.
Sir, audio is not clear. I'm also your PPT is not.
Uh,
Not clear, sir. Audio is not clear.
and one time video. Immediately. Despite
Now it is just visible. Once again, it will be. Okay.
the video is clear, sir? No. Hello.
Now your PPT is visible, sir.
So is this audible and visible now?
Now it is.
Ah, great.
Audio is not clear.
Okay, great.
Now it is clear.
Okay, so okay. Okay. So what happens is the limitations of technology that we can have. Anyway, so in our biomedical lab, we are doing a lot of simulation studies with some funding from exterior and international agencies as well. So eventually, we have ventured into environmental aspects of biomedical research. And during this last 4 years, we have able to develop a strategy to which we can standardize various kinds of molecular aspects and to analyze their propensity to identify false positives and false positive negatives. So this process we put it a patent and anticipating that eventually we will monetize our product also. And we are in talk with some small scale agencies, as you have said, to monetize the product and do some tests as well. So by saying so, I'd like and the things go well. And we are going to put an interdisciplinary aspect to it. And I'm very much thankful to ATAL to give such an excellent platform where interdisciplinary discussion is able and a huge scope of outreach and collaboration is also explored. By saying so, I'd also like to have some question. I'll see if I can answer them. I also thank the organizer for giving me such an excellent opportunity.
Sir.
Yes, I was audible. Hello.
Sir.
Okay. So if there are some questions, they can put it in the chat box as well.
So, participant, any queries, any questions you have to ask or you have to type them in the chat box. Participants.
Thankful to the participants also. Everyone has not left. It is also a good sign.
Any queries?
Sir, how much time does it take to grow the algae?
Sir. Sir.
Sir.
Sir, just ask the question.
Yes, sir. You can put it in the chat box.
No, directly asking the question, sir. Just
I could not. No, I could not hear it. So please request you to put it on the chat box so that I can hear.
Madam, just type the your query question on the chat box. Because of his having some problem on audio.
Yes, I could not hear the question.
Sir is asking that how much time does it take to grow the algae?
Okay. Okay. Great. Yes, great question, sir. Acha. So what has happened since we have isolated these algae from natural resources? So it has been seen that the doubling time of these algae are approximately 28 to 32 in proper sunlight, aeration, carbon dioxide saturation, as well as a particular pH. Okay, but here there is a problem. The problem is you cannot really predict the nature of the wastewater, isn't it? If you are doing, if you are providing some very good resources to grow the organism, it is fine. But in natural habitat, when you are really using wastewater, you cannot able to predict it. So what happens if at all you are seeing that the doubling time is very less? That means very frequently it is growing, but you will not able to exactly say what will happen when you will put it into the real life situation. So there is also a very big challenge. It may also happen that you will find out some algae which takes a lot of time to grow, but once they grow, once they grow, they'll accumulate a lot of biodiesel. So in this way, what will happen is there are two aspects of it. One, you can have a very good growing organism. Another, you can have a low growing organism but accumulates a lot of stuff. So in this way, we have to standardize it. In this.
Any questions, just type it on the chat box. That will be answered by the resource person. Any other queries regarding the session, the topic? Then I stop here. No questions on the side of participants. Sir, you are already in the chat box.
I think one question came.
Just answer it.
Okay. Now, aquaculture is a different thing. It is definitely possible. Why not? But again, then your one objective will go just like that. In aquaculture, what you do is you generate some varieties of shellfish or pearl or like fish and all such things. So it may happen that they'll eat up all your organism. I do not know exactly, but that is for sure because it is also water. That is also water. You can definitely able to do it. But while doing, but one problem is there. One objective is not very this thing because you are using wastewater. In wastewater, if you put your fish, the fish may become toxic or the fish may not be very well growing or it may die. So these are two different aspects. Okay. So I do not know that whether it will be beneficial or not, but we are not looking into that aspect right now. Yes, if not huge, a very good land resource is always very good. There's this Abu Dhabi people, there is huge barren land. They are putting their system there, and if you have huge land, But if you do not very good sunlight, that is also of no use. So two things: one, very good land as well as very good sunlight. Both is needed. Yes, we are there to analyze those things: techno-economic feasibility, as I have said. One thing is very sure that if you can deploy the whole strategy the way we want it, you can get rid of using huge centrifugation and huge amount of fossil fuel in order to generate it. So there are various aspects to it. Techno-feasibility, that is what we are trying to do. We are trying to reduce the use of fossil fuel, that centrifugation, and replace it with an organism, which is very easy to grow, as well as the genetic engineering is also less costly. Let me give you one example. So if I get a funding of 25 lakhs, we can able to generate the lab scale this thing, a fully grown system where wastewater treatment in one hand and biodiesel production in one hand. Biodiesel truly not the diesel, but the lubricant. Okay, 25 lakhs will be enough. Subsequently, for scaling it up, you may require some huge investment. There is no doubt in that. But initially, it can be done in this 25 lakhs. Understood? And you can also able to generate a lot of this thing also. What do you call as it is also very much shall I say you can able to also generate a lot of intellectual property in the process, and it is definitely an environmentally healthy process, and that is why we are talking about it. So what you are doing, you are recycling the environment, and in order to grow the in which aspect you are growing the algae, that is important. Okay, let me tell you this specifically, since you have asked it. I just categorically speak it. Is it environmental friendly? Okay. Let me answer you nicely. So what happens is, where exactly your algae is growing, that is very important. For example, if the algae is growing in a pond where you are trying to put aquaculture, so what will happen? The algae will eat up all the oxygen. Okay, algae will require oxygen as well as carbon dioxide both. So it will end up all your oxygen, and eventually your biological and the chemical oxygen demand will be increased, and so all the fishes will die. Understood? But if you are trying to produce algae for making a biodiesel, so it is very good. So there are two aspects of it. Where exactly the algae is growing, that will decide whether it is that will decide whether it is eco-friendly or not. In this way, I would like to answer it. Means, in general, nobody can answer it, because it may happen that you are doing it for a better purpose. Then it is good. It is not for this, then it is bad. Simple.
Any queries? Otherwise,
Thank you. Thank you.
thank you, sir. Very much for
Yes. Okay. Okay.
Thank you, all the participants.
Yes.
Tomorrow,
Okay. Thanks a lot.
all the participants, tomorrow 9:30 session 4 will be started. Thank you.